Welcome back. This is Coach Heichel. The last two times we've talked about the structure of DNA and the actual replication process of DNA. Now we're going to talk about what RNA is and how it makes proteins and our ribosomes. So the R in RNA is just like the D in DNA. The R stands for ribose, okay? It's the same shape as a sugar in DNA, but now it's called ribose instead of deoxyribose. The N and A still mean a nucleic acid. So let's go back and review what makes up a nucleic acid. And we learned that nucleic acids or nucleotides are made up of repeating nucleotides. A nucleotide consists of three parts. It consists of a sugar. In this case, it happens to be ribose. These circles, if you remember, are going to be your phosphate groups. And these right here, these little gold, marigold, whatever color you want to call them, are going to be your nitrogen bases. Just like in DNA, everything's going to be very, very similar. It's going to be the same shape, same colors, so on and so forth. The only thing that's going to change is that instead of having an A to T like we did in DNA, now we have an A to U. So our C's and G's are still going to be paired up together, and they're going to be paired up with 3 hydrogen bonds. But now instead of having A to T, we have A to U. The big difference. This is DNA, this is RNA, okay? A to U is the big difference when we're talking about DNA and RNA. The main function of RNA is to help our body make proteins. Everything we have, everything we do, is just a different sequence of proteins that genetically makes us who we are. And proteins consist of different types. There's 3 types of RNA. There's messenger RNA and transfer RNA are going to be the main 2 you need to know. There's also ribosomal RNA, which that is just the proteins that are responsible for making the RNA. You don't need to know that for this class. So no messenger RNA and transfer RNA. So when we're looking at a 2-dimensional figure of RNA, there's a lot of similarities to DNA. Okay? We're still going to have A's, we're still going to have C's, we're still going to have G's, but instead of T's, we're going to have U's. Those are going to be our nitrogen bases. The sugars are still going to be the same, and the phosphates are still going to be the same. And it's going to alternate sugar, phosphate, sugar, phosphate, sugar to a phosphate, sugar to a phosphate. Okay? Everything's still going to be the same, except for there is no T in RNA. So no T in RNA. Instead of a T, you're going to have a U. Make sure you understand. No T in RNA. Okay, so protein synthesis is the making of proteins. It's how proteins are actually made. And the sequence of our proteins are what makes us unique as individuals. So it's a 2-step process. The first step is what's called transcription, which transcription simply is making a messenger RNA or mRNA copy of an original DNA strand. This is going to happen inside the nucleus because that's where the DNA is located at. Because we're going to be copying a nucleus, it's going to occur we're going to copy DNA, so it's going to occur in the nucleus. So transcription is just rewriting DNA into an mRNA copy. Once this happens, then the RNA molecule can leave and go to the ribosome because that's where the proteins are actually going to be made at. DNA contains volumes of information about making a protein, but the DNA molecule itself is too large to leave the nucleus, so it has to be rewritten into a language that can be transferred to the ribosome that the ribosome understands, and it does this through mRNA. So this is what happens. The RNA polymerase is going to unzip the DNA molecule at the site of the gene. It's going to be very similar to DNA replication, but now it's just the polymerase is going to unzip it, and then the polymerase is then going to bind the DNA molecule and pair it with mRNA nucleotides. So when we were replicating DNA, A went to T and C went to G. Now that we're replicating RNA, A is going to go to a U, and C and G are still going to be paired together like they were in DNA. Then the DNA is going to re-zip itself together. The mRNA is going to leave the nucleus. It's important that the DNA rezips itself so that it can repeat the process over and over and over again. In case there are some errors, it can go back and fix itself. Once it's in the ribosome, okay? Once it's in the ribosome, a process called translation is going to begin. And this is when amino acids are going to be assembled actually into the proteins that makes you and I unique. And the way translation works is mRNA is going to enter the ribosome. It's going to pair up with tRNA. Okay, 3 mRNA nucleotides are going to be called codons, which are going to be paired up with anticodons, which are tRNA. Amino acids are going to be added until you receive a stop message. On your codon chart, there's going to be 3 stop messages or 3 stop codons, which is going to tell the ribosome to stop making that protein at that specific moment. So it's also important to know that a codon, again, is 3 mRNA bases. An anticodon is 3 tRNA bases. 1 codon makes 1 amino acid. So every amino acid consists of 3 letters. For example, A, U, G. That would be 1 amino acid. Make sense? Great. Let's keep going. So here we go. We got our DNA strand. It's going to make RNA. This part right here, this first step, right here, this is going to occur in the nucleus. Okay? It's going to happen in the nucleus because we're copying DNA, and that's where DNA is located at. So the mRNA. Remember, A's go to U's, C's and G's go together. So this T is going to be an A. This A is going to be a U. This C is going to be a G. So on and so forth, all the way down the line. Okay? Makes sense? This U came from this A. This G is the complement, the C. The C complements the G. CCA complements the GGG. So on and so forth, all the way down the line. And continued more on the next page. So now that the mRNA is made, it's going to leave, and this little thing is going to hightail it out and go to the ribosome. Once it's in the ribosome, I forgot my O there. Once it goes to the ribosome, then it's going to make amino acids. But before it does that, it's going to attach to tRNA. tRNA and DNA are going to be identical, but wherever you see a T, you're going to replace it now with a U. So TAC is going to become UAC. AGG is going to become, guess what? It's going to become AGG because it's identical. The only difference is wherever you see a T, you're going to now put a U. Okay? So GGT is going to be GGU because there is no T in RNA. Wherever you see a T, you put a U. So on and so forth. All of your examples if you want to fill those in. So now that we have our tRNA and our mRNA, we're going to use what's called an amino acid chart. And we're going to use mRNA. On your amino acid chart that's being passed out, you should write use mRNA. Okay? You're going to have 1 or 2. You're going to have one that looks like this, which to me is the easiest, but I'm going to teach you both so you know how to use them. If you're using this one, okay, and for example, you want to find the amino acid, you have GAC, okay? You're going to start with this big G right here in the middle, okay? You're going to start with this big G. Then you're going to go out to the A. You're going to come out to this A right here, and then you're going to go out to the C, and your protein that you just made, or amino acid, is going to be aspartic acid. Makes sense? So what would UUA be on your circle chart? If you're making an amino acid, UUA would be what? Okay, great. So let's start at this U. Let's go out to this U. Let's go out to this A. And your amino acid you made from UUA is going to be leucine. Okay, some people like this chart over the circle chart. It's going to work the exact same way, except it's kind of like a multiplication table. If we were going to go UUA, since we're familiar with UUA, you're going to start at this U, you're going to go over to this U, and then you're going to find the letter that corresponds with the third base over here. So A, we're going to come back, U, U, A, we're at leucine again. Great. So here's our 3 stop codons. They would be a U, a U, a G, and our last one is a U, G, A. And you only have one methionine, which is going to be AUG. Methionine, beside methionine, go ahead and write at your start codon That means the protein won't be made until that start codon's received. Once that start codon's received, all these letter combinations are going to be read until it gets to one of these three stops. Then it's just going to stop making it. And I'll show you an example coming up. So we have this, we have our mRNA, we have our tRNA. So we're going to make our amino acids now. Well, AUG, we know is methionine, which is the same thing as start. So now the protein's going to start being made. Methionine is not actually going to be part of the protein. It's just going to give the ribosome the signal or the green light to start making it. So it's going to keep going along. And UCC is going to be serine. CCA is going to be proline. UGC is going to be cysteine. GGC is going to be glycine. UUA is leucine. 3 C's in a row is going to be proline. UAA is stop. Stop. So this protein that the ribosome just made for us is 1, 2, 3, 4, 5, 6 amino acids long, has 18 bases, because each base or each amino acid has 3 bases or 3 letters. Okay, so we have 18 total bases for these 6 proteins that were just made. Now there are more on that other page, but since we got to that stop codon, that's where we stopped making it at. So just a brief compare DNA to RNA type deal. DNA, the sugar is deoxyribose, okay? RNA has a sugar that is a ribose. Both of them are going to have adenine because both of them have an A. Both of them are going to have cytosine because both of them have a C. Guanine is going to be present in both. Thymine is only going to exist in DNA. Uracil is only going to exist in RNA. DNA is a double helix. RNA is going to be single-stranded. DNA is located in the nucleus. Both of them are composed of nucleotides. DNA stores your genetic information, and RNA is going to help make your proteins for you. And until next time, if you have any questions, please feel free to email me or ask. Thank you. Have a wonderful day.